Dear students, we will have a lecture on spectrophotometer, that is an instrument used in biological laboratories. What is its principle, how it works, what are its different parts, and how it is useful to the biological laboratory. We'll learn in coming slides. Spectrophotometer is an instrument that measures the amount of light absorbed by a sample. Our spectrophotometer techniques are mostly used to measure the concentration of the solutes in solution by measuring the amount of light that is absorbed by the solution in a cuvette placed in the spectrophotometer. Arnold J. Beckman and his colleagues at the National Technological Laboratory invented the Beckman DU spectrophotometer in 1940. Spectrophotometer is basically a system, instrument in which there is a light source, then a lens or that is a collimator which is passing the light through a monochromator, that is a prism or grating, and the 7-color spectrum is created after passing through the prism or grating. The wavelength selector selects the wavelength whatever is needed for that particular experiment is passed through the sample solution present in the cuvette, and finally the photocell detects the absorbance or transmittance on the screen. So it is the line diagram of the instrument given in the slide. The spectrophotometer technique is to measure the light intensity as the function of wavelength. It does this by diffracting the light beam into a spectrum of wavelengths, detecting the intensity with the charge coupled device, and displaying the results as a graph on the detector and then on the display device. The spectrophotometer is having prism or grating which is used to split the incident beam into different wavelengths. By suitable mechanisms, waves of specific wavelengths can be manipulated to fall on the test solution. The range of the wavelengths of the incident light can be low as 1 to 2 nanometer. The spectrophotometer is useful for measuring the absorption spectrum of a compound, that is, the absorption of light by a solution at each wavelength. The spectrophotometer, the instrumentation, that is, the essential component of the spectrophotometer includes a table and the cheap radiant energy source, then material that can be excited to high energy states by high voltage electric discharge or by electric heating, serves an excellent radiant energy source, a monochromator to break the polychromatic radiation into component wavelength or bands of wavelength. The monochromator resolves the polychromatic radiation into wavelength into very narrow bands. Prism, a prism disperses polychromatic light from the source into its constituent wavelength by virtue of its ability to reflect different wavelength to a different extent. Two types of prism are usually employed in commercial instruments, namely 600 Cornu quartz prism and 300 Litor prism. Grating are often used in monochromators of the spectrophotometer operating ultraviolet, visible and infrared regions. Otherwise, we can use prism for the visible wavelength light, but grating is generally used for ultraviolet and infrared as well as visible light. The transport vehicles to hold the sample is called as cuvettes. Sample to be studied in the ultraviolet or visible region are usually glasses solution and are put in the cells called as cuvettes. Cuvettes means the visible region are made up of either ordinary glass or sometimes quartz. The photosensitive detector and an associated readout system. Most detectors depends on the photoelectrical effect. The current is then proportional to the light intensity and therefore a measure of it. Radiation detector generate electronic signals which are proportional to the transmitter light. These signals need to be translated into a form that is easy to interpret. This is accomplished by using amplifiers, ammeters, potentiometers and potentiometric recorders. Some of the major applications of the spectrophotometer includes the following: the detection of concentration of substances, detection of impurities, structural elucidation of organic compounds, monitoring dissolved oxygen content in fresh water and marine ecosystem, characterization of proteins, detection of functional groups, respiratory gas analysis in hospitals, molecular weight determination of compounds. Visible and UV spectrophotometer may be used to identify classes of compounds in both the pure state and in biological preparations. The spectrophotometer thus works on Beer's and Lambert's law, that is, wavelength is directly proportional to the concentration and the length of path, or we can say the lambda is proportional to C and L. So, spectrophotometer is very important instrument for quantification of some of the materials, that is phenols, dihydroxy orthophenols and the proteins, and we can have the system like spectrophotometer which determines or quantify the amount of protein or some other compounds in a solution and have a graph for that, by which we can analyze any standard system for the applications in the biological system. Thank you very much.